Good morning, everyone. Today we are going to study about new drug discovery process. Drug discovery and development is a challenging and expensive activity of the pharmaceutical industry. The aim of the new drug discovery are to develop clinically efficacious and safer drugs, economically viable, discover entirely new class of drugs, or to discover newer clinical uses of older drugs. Explore the mechanism. For every 6,200 compounds synthesized, only 21 will reach the testing stage for subacute toxicity. Only 2 to 6 of these compounds will reach the clinical trial and subsequent evaluation. One of these compounds may become a drug. Birth of a new drug. This is the birth of a new drug. This is sources, synthetic synthesis, then biological screening, blind random and activity, then clinical trials. This is all that schematic representation of a new drug development process. New drug development, drug discovery. The discovery of new drug and their development into commercial product take place across the broad scope of the pharmaceutical industry and research institute. The combined efforts of chemists, biologists, pharmacologists, toxicologists, statisticians, clinicians, pharmacists, and pharmaceutical scientists, engineers, and many others are involved in the drug discovery and development process. The source of new drugs may be from a variety of natural sources, that is plants, minerals, and marine, or chemically synthesized in the laboratory. Throughout medical history, plant materials have served as a reservoir of potential new drugs. After the isolation and structural identification of active plant constituents, organic chemists may recreate them by total synthesis in the laboratory or, more importantly, use the natural chemical as a starting material in the creation of slightly different chemical structures through molecular manipulation procedures. The new structures, called semisynthetic drugs, may have slightly or vastly different pharmacological activity than the starting substance. The advent of genetic engineering and some microscopic DNA manipulation techniques, the development of pharmaceutical products, has witnessed a new era. There are two basic technological advents that drive the genetic field of drug development: recombinant DNA and monoclonal antibodies. The recombinant DNA can potentially produce almost any protein, which may serve as model for the creation of new drug molecules. Both these technologies are greatly contributing to the human gene therapy, which is based on the modification of the genetic material of living cells to prevent, treat, cure, diagnose, or mitigate human disease. Methods of drug discovery: random or non-targeted screening. It involves the testing of large number of synthetic organic compounds or substances of natural origin for the purpose of detecting biological activity. Random screens may be employed initially as a means to detect an unknown activity of the test compound or substance and identify the most promising compound, to be studied by more sophisticated targeted screens to determine more specific activity. To detect and evaluate biological activity, bioassays are used to differentiate the effect and potency of the test agent compared to control of known actions and effects. The initial screens may be performed in vitro using cell cultures to test the new agent's effect against enzymes, systems, or tumor cells. Molecular modification involves the chemical alteration of a known and previously characterized organic compound for the purpose of enhancing its usefulness as a drug. This may enhance its specificity for a particular body target site, increase its potency, improve its rate and extent of absorption, modify its pharmacokinetic in the body, reduce its toxicity or change or other physico chemical properties, knowledge of chemical structure, pharmacological activity relationship. High-throughput screening: the conventional bioassays and serendipitous discovery of new drugs were extremely time-consuming, expensive, and required large amounts of the compound. This technology not only provided a rapid means to evaluate small amounts of compound directly for the ability to interact with a receptor or enzyme, but also helped to develop radioligand binding assays for large number receptors, their subtypes, and enzymes. The information derived from these studies is invaluable in the design and synthesis of more potent and target-selective analogs. The simultaneous evolution of computer-based molecular modeling and characterization of three-dimensional structures of many pharmacologically relevant proteins. The design of molecules that interacted with these biological targets has moved the drug discovery process from an irrational fit and missed strategy to the rational approach to drug discovery. Several innovations in filtration technique, miniaturization, and automation in binding studies during the last two decades have made it possible to screen 50,000 to a staggering 2 million compounds. The most efficient high-throughput screening assay systems include mini-miniaturized samples well to allow the handling of more samples at any given time. Mechanism-based drug design involves molecular modification in designing a drug that interfaces specifically with the known or suspected biochemical pathway or mechanism of a disease process. The intention is the interaction of the drug with specific cell receptor enzyme system or the metabolic process of pathogen or tumor cell, resulting in a blocking, disruption, or reversal of the disease process. Designing drug on the basis, therefore, it is essential to understand the biochemical basis of the disease process and the manner in which it is regulated.